Hello everyone, so my name is Yuni Lei, and I will be talking on gene editing of human embryonic stem cells via an engineered microbiofactor carrying zinc finger nucleases. So here's an outline for today's talk. We'll start with an introduction on what is inspired us to investigating this story. Then we'll talk about in the results section, demonstrating the capability of our system. And finally, I would like to conclude on what we think the outlook of the gene therapy field. So first, the need for gene therapy. Genetic disorder is defined by an illness caused by abnormality in genes or chromosomes. One example of the genetic disease is X-linked severe combined immunodeficiency, or most people know it as the bubble boy syndrome. The prevalence of the skin disease is around 1 in 100,000 births. Essentially, it's a mutation in the R-gamma common receptor genes of the IL-2, which that results in a defective T or B cells. Essentially, this mutation alone will wipe out your whole immune system. The victims are extremely vulnerable to infectious disease. If untreated, the children usually die within a year due to severe recurrent infections. So, since this is a mutation in a specific genes, this is an ideal target for gene therapy. And that's what people have done. In year 2000, 2 independent groups actually tried to use a gene therapy technique to cure the disease. What they have done is they took out the stem cells from those SCID patients. They enrich the stem cell, transduce them with a retroviral factor that carry a functional therapeutical genes, in this case the IL-2 genes, and then they reinfuse the treated cells back to the patient. And they found that most of the patients respond to the treatment, meaning they were healthy from the skin disease. However, later on, they found out that these treated patients developed leukemia after the treatment. So then that leads us to investigate what was the underlying mechanism for causing the leukemia. So people have found that the nature of the retroviral factor has a tendency to integrate its genomes into the enhancer, promoter, or within the genes. And these insertions actually cause an activation of oncogenes. Essentially, those treated cells become tumorigenic. So that leads to how to safely design a viral vector system in which we can introduce the genes only to the target genes without disrupting other healthy cells. So what people have found is using a so-called zinc finger nucleus. These zinc finger nucleus are designed so that they can bind to the target genes, in this case an IL-2 genes, generate this double-strand break. While if we supply a donor template that has a homologous sequence to the disease genes, then the homologous recombination, a naturally occurred mechanism, will take place. We'll swap out the defective genes with the healthy genes. And the end result is you have a functional genes and without any of the viral vector introduction. So we have the tools needed to genetically modify the targets of the disease cells. Then we need to look at what are the potential target for the treatment. What we have been looking at is the embryonic stem cell. They have two distinguished properties from other cell types. First, they can self-renew indefinitely, meaning once you treat the cells, then you can have a lifelong supply of that treated cells. And they can also be induced to become any of the cells within your body type. So what we are hoping to do is we modify the ES cells in vitro with our zinc finger nucleus system. So they will genetically modify those disease genes with the healthy genes. And then we will then infuse back those treated cells to the patient. Ideally, this treatment will turn those mutations back to the original correct sequence. So then we look back into the literature to see what people have done in the past. Two groups came out. The Lombardo group published a paper in 2007 with a HIV-based gene delivery system. So they encode the zinc fingers along with a donor template in the HIV-1 viral vector. And then they transduced the embryonic stem cell. And then they were able to show that up to about 5% of the treated cells are GFP positive, are genetically modified. However, this system, one of the downsides of the system is since it's HIV-1 based viral vector, it has a possibility of generating the recomplication recombinant virus. So in 2009, another group utilized a virus-free delivery. They were using a similar technique, delivering the zinc finger nucleus, and then they were able to show that about 0.2% of the embryonic stem cell were genetically modified. This is a more promising result in the gene therapy field. However, the efficiency was much lower, 20-fold lower than the viral vector system. So that makes us want to investigate more on how to make a system that's capable of giving a high efficiency and a low safety concern. So here is a candidate that we found for gene delivery vehicle. What we have been using is baculovirus. Essentially, it's an insect virus. And then it's known to not replicate in mammalian cells, but the virus is capable of transducing mammalian cells. So the idea is this system will work similar as a viral vector, but it doesn't have all the side effects of viral vector, meaning the generating of the replication competent virus. The target gene that we'll be testing in our system is a human CCR gene. As people have found that homologous null mutation is actually prevalent in a small population of individuals. And the disruption of this gene is well tolerated. And also, the gene inserted into the CCR locus can be actively transcribed. So here is the result on our system. So what we have shown here is our target site, the CCR locus. So we are using a baculovirus to deliver both the zinc finger nucleus and the donor template into the target cells. And the zinc finger nucleus will bind to the CCR locus, generate this double-strand break. And when we have this homologous region to the, we have the donor plasmid, the donor plasmid that has this homologous region to the CCR locus. And then the naturally occurring homologous recombination will take place to swap, to insert this system into the CCR locus. And we have demonstrated that this in two different immortalized cell lines, showing that with the introduction of the donor DNA and the zinc finger nucleus, we can turn the cell in green. Whereas by donor's DNA itself, there was no signal detected. So before we want to move into the embryonic stem cell, we want to further optimize the system. So we did a dosage experiment, and we found that we can correct up to 10% of the immortalized cell line. So then we did the same experiment in an embryonic stem cell. And we can, as you can see here, we can correct up to 4% of the embryonic stem cell. This efficiency is roughly similar to the HIV-1 based viral vector. And then we also demonstrate that by using different antibodies staining the treated cell, that we can show that these treated cells remain undifferentiated by using these four commonly known markers for the embryonic stem cell. And that those stem cells are fluorescent green, meaning that these cells are treated by our zinc-finger nucleus and they are genetically modified. So finally, I would like to talk about what we think the future of the gene therapy field holds. It's very promising. It's a very promising technique to treat many incurable diseases, especially for those genetically inherited disease. But so far there are two important factors that must be considered. The first one is we need to find more therapeutically suitable genes. And secondly, we need to further optimize the gene delivery vehicle. So what I have been talking so far is based on the cell therapy. What we have to do is we have to take out the cells from the patient, individually modify it, and then we plant it back to the patient for further treatment. However, if we can design a vector in such a way that the vector can target to the specific cell type within the human body, then essentially we can think of it just one injection. Then the vector will bind to the target cells. For the skin disease, it's the hematopoietic stem cell. And then once that's occurred, then we can perform this recombination to fix the defective genes. So that is an area that our lab is actively investigated. And we'll have two posters in this afternoon, one talking on the modification of the delivery vehicle. One by Laura Liu on site-specific modification of AAV2, and one by William Hu on the IDOV targeting to dendritic cells. And finally, I would like to thank my lab mates and my collaborator, Dr. Victoria Fox from Stanford Core, and our founding source, the California Institute for Eugenic Medicine.
There's time for questions. I have one. All right, so presumably you're growing up your baculovirus in insect cell. So given that the baculovirus is grown up in insect cells, I'm wondering if there's any danger of contamination from the insect cell culture.
So meaning that there is foreign proteins from the insect cell. Well, before injection, we also always do a purification process. So we purify out only the viral vector that we're using for any of the treatment. And most of the protein, or actually most of the viral vectors, are inactivated in the mammalian cell system. So chances are there's not many concern coming from the viral vector, but there might be some protein that's coming from the insect cells that might potentially cause a problem.
Any other questions? All right, let's take it out once more.